Hi there. So we're gonna start a new series of videos about photosynthesis. So we're right here in the scheme of all the topics we're gonna cover in the class. Photosynthesis is very special to me because I did, until I started teaching this class a few years ago, I had no idea how beautiful. Of course, everybody knows it's important, but how beautiful it is. Because I did almost all my training in medical schools, and I guess you don't see many plants walking into the operating room, and the topic is not covered. But it is covered here, and that's a good thing, as you will see, hopefully. So in this video, we're going to just go first through some basic properties, and in subsequent videos, we're going to see more complex. I mean, in more detail, the kinds of photosynthesis that we have, photophosphorylation, and how we actually make how plants make sugars, the sugars they need from basically thin air, from CO2. And we're going to close with a Calvin cycle on this topic. All right, so you've seen this slide before. This is the central way of looking of energy and chemistry and chemical transformations on this planet. Who pays for almost everything that we do in the biosphere? It's really the sun. Okay, this is where the energy is coming from. We've discussed many times that cells operate far from equilibrium. So there is a huge thermodynamic cost, and the source of energy, the one who pays, as I said, for all this is the sun. But for that to happen, somebody has to capture that energy from the sun. And that someone is, of course, the green things on here, the phototrophs that can synthesize their food essentially from light. That's what that they feed off the light. That's what a phototroph means. And in doing so, of course, they make a lot of themselves, which is made of organic compounds, and that can then serve as a food for organisms like us, which we eat them, and we eat others that eat phototrophs. And everybody does the same thing, and then we go around and around. And this is really how the radiant energy from the sun gets captured in chemical energy in the biosphere. And ultimately, of course, the entropy increases, but not in our cells. Entropy increases in space and in the whole universe. So it is this process that we are gonna be dealing with, and how from the sun, how the light gets, how the radiant energy is captured, and how that is used to in the form of chemical energy to synthesize organic compounds. So it turns out that all phototrophs are capable of chemotrophic metabolism. So all of them, all green things, the ancestor of all green things was a red thing at some point. And in early life on Earth, billions of years ago, probably they could get some of that energy from whatever was around, minerals and sources of energy, volcanic, usually a volcanic source. To this day, actually, deep down in the ocean, in the vents, where the crust is essentially open and some material flows out into the ocean, you do find bacteria that can do this kind of thing and do not actually depend on light to synthesize the things they need to. But for almost everything else, the ones that do photosynthesis, for example, they were always all the chemotrophic metabolism, everything we have discussed up to this point, they can do this, all of it, which suggests that photosynthesis was an add-on. And the way to see photosynthesis is really the reverse of respiration that we just discussed. In respiration, we had oxidative decarboxylation of organic substrates, right? We lost the CO2, let's say in the TCA cycle. Well, what happened, and we took out, the electrons, so that they found their way onto NADH, right? And which will then fed into respiration, and we got ATP. But during that process in the TCA cycle, electrons were sucked out of these compounds, of these substrates, and they got decarboxylated. Okay, so in photosynthesis, we have the opposite. We have reductive carboxylation of organic substrates. So we start with something that gets carboxylated in the form of CO2, and that something gets reduced. Okay, so the organism provides the electrons, the reducing power to reduce those fully oxidized carbons that have been added on to other substrates. So photosynthesis is actually a compound word, and that's a good thing because biologically it's also split in the photo part, the light part, and the synthesis part. In the light part, we literally have the capture of the radiant energy, light, and the cells convert that energy into chemical energy in the form of ATP and NADPH. Now we start seeing NADPH. Okay, we haven't seen NADPH up to this point. And if you remember back in the introduction, we said how we see NAD and NADH in catabolism, but we see NADPH in biosynthesis, in anabolism. And this is where we're going to start seeing that. In the synthesis part, in the other part after the photo part, CO2 and the chemical energy that we generate in the photo part, the light part of photosynthesis, now can be combined to give us sugars. Right? So CO2 is reduced to the level of carbons in the sugars, so that was hydroxycarbon. Okay. The general reaction is like this: CO2 plus some donor. Usually is water, but it doesn't have to be, and in some cases it's not. Green plants, in the case of green plants, it is water, light, and then we have the larger sugar shown here. As I said, the donor doesn't always have to be water. Overall, in green plants, this is how it works. Okay, so it is water, the donor, and we have CO2 plus water, which will provide the electrons, and then with light, that gets converted to glucose, let's say. And in the process, as a byproduct, we get oxygens that came from the splitting of water. So a schematic then is like this: we have light, water, and we don't have any CO2 in this part. So in the light reaction, we just have light and the source of electrons. And what's gonna happen? The electrons are gonna be transferred onto ATP and NADPH, on NADPH, and we're gonna get ATP also. Okay. So the electrons from water are gonna reduce NADP plus to give us NADPH. These are very powerful, reducing power. A lot of it that the photosynthetic cell is able to synthesize. And in the process also, we're going to see the formation of ATP. Now, we're going to have some electron transfer here. So electron transport chain will come into play again, just like we saw in the previous videos during respiration. And yet, as the electrons are moving from photosynthesis, from water, those electrons, they will find their way eventually onto NADP plus to give you NADPH. But in the process, as you can guess already, okay, they're going to trigger some localization of protons into one compartment of a membrane versus the other, and that will be then able to drive ATP synthesis. Now, a byproduct of this splitting, of water is the generation of oxygen from the light reactions. Now, the sun goes down. What happens at night? Well, at night, what happens is CO2 comes in, okay, and the weight is added to a precursor substrate. Is going to be described in the Calvin cycle, but you use this chemical energy that you've made from the photo part to fix literally CO2 and reduce it, okay, to the level of sugars. So you're going to end up with sugar. And of course, in the process, you generate NADP plus, and you've spent your ATP. How much sugar are we making, or how much of these substrates in general? Okay, let's start with CO2. How much is fixed, for example? So we get CO2 coming in, oxygen out, sugar out. How much of that stuff is made every year? Okay, a huge amount, as it turns out. And the easiest one to visualize is when we talk about sugar. So let's start with this. Okay, how much sugar is made every year on average on this planet from photosynthesis? It's a giant heap of sugar, and the base of that heap is 43 square miles. Just for reference, from College Station to Houston, it's about 90 miles, so halfway there. Imagine a base that's 43 square miles, so 43 by 43. and the heap is as high as 2 miles. So it's a giant mountain, essentially, of sugar is made every year. In the process, this much, tons and tons, millions and millions of tons of CO2 is fixed. Half of it is in the sea and half of it is on land. And a lot of oxygen, about the same volume here, or weight is released. Tons of oxygen is released every year in the atmosphere. The free oxygen, as it turns out, it turns over every 2,000 years. Now, again, if all photosynthesis stops, all life will end in 25 years or so. So we have gas exchanges here, okay? So we have absorption of CO2, we have generation of oxygen, and that became clear to Joseph Priestley, who actually saw that and proposed it in the late 1700s. And a few years later, Ingenhouse actually specifically said that plants make oxygen in the day, and they also make CO2 at night. Now, yes, they absorb CO2, but they also do other things at night, right? So they do their TCA cycle, and all that stuff is happening, so they can actually release CO2 also. They don't just absorb, right? So that's important to remember. The plants still do everything else that we do. Okay? They just have an extra thing that they can do, which is photosynthesis, which we cannot do. Since we're talking about light, some basics, just so that we're on the same page. These are the various wavelengths of the various radiations that we've all heard about. The more you are on the left, the higher the energy. The more you are on the right, the lower the energy. Longer wavelength here, you can see how the frequency changes here, and short wavelengths, on the left. So we're concerned only with the visible part of the spectrum here, which is this tiny, tiny little sliver there. And those are the colors of the rainbow. And again, short wavelength, close to UV, close to ultraviolet, have the highest energy. And then we have the longer towards the red. Redshifts, they're longer wavelengths, lower energy, as you'll see. Now, we're talking about energy, but how is that described? But of course, the most famous equation that everybody knows. And that gives you the energy of a photon, E, okay, equals the frequency of the radiation and times the constant, Planck's constant. So that's, of course, the famous Einstein equation. And since we were dealing with energy, let's convert it to something that we have already seen in our discussion here about metabolism, metabolic reactions, let's say kilocalories per mole or something, right? So for that, we have to get to the mole. So a mole of photons the unit is called an Einstein. So a mole of photons, then all we have to do is just multiply this product of Planck's constant with frequency by the Avogadro's number, shown here with n. And then we can actually get some real numbers. And the velocity, now frequency, of course, is velocity over wavelength. And this is where the wavelength comes into place. So the energy will be lower at the longer wavelength because the denominator gets bigger, right? So we have 700 nanometers. That's a longer wavelength, and so that will drop your energy. So by almost half, actually. So you have this is the visible part of the spectrum where the business of photosynthesis is happening. You are at close to the red part here, the 700 nanometers. You're going to get 41 kilocalories from 1 mole of photons. If you are at a shorter wavelength, at 400 nanometers, you're going to get 72 kilocalories per mole of photons. So there is a big difference. Again, reflecting the point here that short wavelengths, high energy. So fine, that's all good. Those are the equations and the numbers. So there is energy in light. This is what they were supposed to convince you, all these slides. But the point is, how do phototropes capture that, right? How do they manage to capture that energy? So they need some special compounds for that. And they do make chlorophyll, which was discovered 200 years ago. And chlorophyll is special. It can capture that. It's photosensitive, so it's got a chlorine ring. All chlorophyll molecules, 2 major ones, chlorophyll A, chlorophyll B. You'll see how they look like in a minute. They have this chlorine ring in them, right? So you have all these nitrogens. It's a kind of a special compound. It's all the nitrogens. You have magnesium in the center of the chlorine ring too. And the name, of course, means green leaf, chlorophyll. It's what makes plants green. And but there are various other side chains to the chlorine ring, and you depending on what you have on the side chains, you get chlorophyll A or chlorophyll B. The only difference here is one extra keto group on chlorophyll B. There is pheophytin. Again, there is a chlorine ring and all the side chains, but they're all these photosensitive compounds that plants have. So what happens when, let's say, light hits an electron? The electron is spinning around the nucleus, perfectly happy there, and then it gets hit by a bolt of light. Then the energy is going to go higher on that electron. It's going to be at an excited state. It's going to spin at a different orbit. And that will not last very long. It will come down. And when it does come down, you have a release of heat. And you have another photon coming out sometimes in the form of fluorescence, right? And again, you can have to imagine a chlorophyll molecule on some leaf, and there is a photon coming from the sun, which then is able to excite this electron. If you look at the amount of light that is absorbed, okay, so they have to absorb this light by the different chlorophyll molecules, so that you have a very specific spectra. Chlorophyll A absorbs, 430, I believe, or something like this, and chlorophyll B is a little longer the wavelength. You have carotenoids, which we didn't discuss now, but we're going to see them later. They also absorb light, and that's again the visible spectrum. To see how much, so this provides now a way to actually see photosynthesis from space, see where chlorophyll is, right? And on a planetary basis. And that's exactly what NASA has done. I'm not going to play the video, but because somehow I tried it and it messes up the presentation. But here's the link. This is what your tax dollars pay for. And in this case, NASA, what they have done is they have put all these telescopes on satellites, and those are sensitive specifically to, or some of these telescopes, specifically to be able to detect the absorption of chlorophyll on the planet. And this is a composite video over many, many years, I think almost a decade, all around, okay? And as it goes, as the Earth spins, so they can capture the entire surface of the planet. And as the seasons change, and you can see here, for example, that there is very little chlorophyll, there is very little photosynthesis happening in the Sahara. Okay, there is not a single green leaf there. But you see a lot in where the forests are. You see a lot in the oceans, okay? As I said, half of photosynthesis is actually happening in the oceans. But the most beautiful part of the video, when you get to see it, and I hope you do, is that it's actually pulsating with the seasons. Okay, there is an oscillation here on the amount of chlorophyll. If it's winter in the northern hemisphere, and there is not much photosynthesis going on, you can see actually this boundary is moving up and then down with the seasons as they go on, which is quite spectacular. It's really the breathing planet, the living planet. To me, it's the best visualization of that concept, that we are living on a living, breathing planet. The other thing you see is again, it's better on the video, but you can actually see it even a little bit here. You can see these massive flares, in the oceans. And you can see this in a few places. Probably the big rivers are coming off here, but you can see in the Sahara, and you ask, okay, what is going on here? Well, what is going on is when there are all these winds that are blowing off, so you have all these algal blooms essentially in the ocean. And how is that happening? Well, when you have some river, you have a delta of some river, that will happen. There is a lot of food, so the algae will grow and they will photosynthesize, and then you see it from space. Or when the wind blows, minerals from the Sahara, which essentially, feed the algal blooms in the Atlantic Ocean, and they go as far as the Amazon to actually fertilize the forest, on another continent across an ocean. It's truly stunning. I mean, they're spectacular videos. I should probably stop talking about them right now because we have other things to cover, but I urge you to go watch them. All right, so in the more mundane slides here, we go, from this beautiful thing to my little scribbles here on the slide. This shows our old favorites, the E values, the electron potential here, and what happens when that electron gets hit by a bolt of light. And again, this is in reverse, so this is negative, this is positive. Sometimes I flip the orientation of the axis, it doesn't matter. But imagine now a low energy electron sitting at some photoreactive center, at some leaf, you have a chlorophyll there, and sitting around happily ever after. It's low energy because it's very positive, okay? And the E value, let's say 1.2 volts, 1 volt or something like that. It sits there and then gets hit by light, okay? The energy is quite substantial, almost a volt. And now that electron will have a very high energy, like it's going to be minus 1 volt, the E value. And just to remind you, the more negative the E value is, the higher the energy of electrons. That's another slide from the very beginning of the class. So light will raise the energy of that electron. And that's measurable. Now, that electron is a very strong reducing agent, very reducing. So what is its fate? Well, it has multiple fates. The excess energy, where would that go? It can just go away as heat. Okay, that's what happens. Okay, so nothing will happen. Just go away as heat. It will fluoresce, and a small fraction is lost that way, actually, in real life. But during photosynthesis, once this electron gets raised to this high energy, then it gets transferred to an adjacent molecule with very little loss in energy. And that will happen as long as the next molecule is very close and the absorption spectra overlap, okay? And then you have this series of acceptors and donors, and eventually finds itself back down to where it was at the ground state. So that's the good fate, okay? This third fate is what we want to capture that energy. Equivalent here is, when burning, if in terms of heat, if you burn your food that you eat right away, and you are not able to capture with little reactions, that energy that we saw, let's say in glycolysis, as opposed to that, you just burn it all the way, like you burn a piece of wood, then you lose all of it in heat. Nothing has been captured, right? But so we want this. And fluorescence is nice, it's beautiful, you see colorful things, but the point is here to capture that energy. So, and the differential, of course, is 1 volt. And if you remember, you need about, maybe somewhere between 200 and 250 millivolts to make 1 ATP. So here, clearly, that differential is enough to make a few ATPs, 3, maybe 4, right? So, where are all these things happening? I mean, it turns out it was discovered almost 100 years after chlorophyll was discovered that chloroplasts are actually similar with cyanobacteria, chloroplasts in plants. And that's a picture of a cell, and you see the chloroplast now here in the cell. I forgot what kind of cell that was. But this little green things in the cell, this is where chlorophyll is now. And so chlorophyll is in chloroplasts. A better view of the chloroplast is here in this schematic. It's an organelle. It's a membrane-bound organelle. So here we are again with membranes. And you have a big sac here, and in it you have the stroma, which has a high pH. Okay, high pH means low concentration of protons. Okay, the lower the pH, the higher the concentration of protons. But within it, you have these stacks. Okay, these are the business part really, and you have the thylakoids, and they stack. And they communicate also with a lamella shown here, and it's the thylakoid. You can just imagine a book stack, and you just let's take one book that's going to be one thylakoid, and you see it. This is how it looks like. That visual is very useful for us to imagine this like the mitochondrial thing that we had seen, except that everything is inside out now. So the orientation is completely opposite from what we had seen in mitochondria. The thylakoid is here, and outside of the thylakoid, This is where the protons are low, okay, because the pH is high, and then light gets hit, and it gets captured by chlorophyll, which is in there in the thylakoids. Water gets split, oxygen comes out, and the electrons gets passed on, as we'll see, eventually to an ADP to become an ADPH. But what happens in the process with that electron that we just discussed is that there is an electron transport chain, very similar to what we saw in mitochondria. And in the process, as the electron moves along the electron transport chain in the thylakoid, in the chloroplast, protons get inside, okay, the thylakoid. So they go into the lumen of the thylakoid. So this is the opposite of what we had seen from the inner mitochondrial matrix. The protons go out. Here they come in, okay? And so you have this amazing gradient, the proton motive force that is built across the thylakoid membrane. You accumulate all these protons inside in the lumen. And of course, they want to go out. I mean, that's how the battery is charged here. So photosynthesis charges a battery in the thylakoid in chloroplast. The protons want to come out, and all you have to do is just stick an ADP synthase right there at the membrane. But again, the orientation is the opposite. Okay, so the soluble part that makes ADP is actually on the outside now, right? So the protons, as they want to come out, they go through ADP synthase, they come out. That's where they want to be, and ADP synthase, of course, makes ATP now. So this is really how the energy is captured, okay, by photosynthesis in kind of in the same way that we had seen in respiration. So a battery is charged, and that is coupled to an ATP generation, the ATP synthase. What is not shown in the schematic again is that in the process also we get reducing power in the form of NDPH. So it's almost certainly the chloroplast that we see today is a relic of the symbiosis phenomenon. Some cyanobacterium was captured at some point by a primordial eukaryote, and eventually became the chloroplast, and everybody gains. That's how the cyanobacterium looks like, and today, okay, and it can do photosynthesis. They were the first organisms that could do photosynthesis, and as I told you, due to these organisms, oxygen started to build up in the atmosphere, billions of years ago, and then everybody had to do oxidative metabolism, oxidative phosphorylation. Something like this happened with and mitochondria were primordial bacteria were captured by eukaryotes, became mitochondria, and primordial cyanobacteria were captured by other eukaryotes, and they became chloroplasts. And everybody gains in the sense that the chloroplast still has some of its DNA in there, just like the mitochondria does, but most of its genome of this primordial or whatever cyanobacterium that was captured, most of these genes now have passed on to the host in the nucleus, and the host is responsible for maintaining them, all that. So it's kind of the same principle. All right, so we're going to stop at this point, and then we're going to see the kinds of photosystems that we have happening in photosynthesizing organisms. So let's stop there.